Hi everyone, I'm Elizabeth McKenna, Executive Editor of Cancer Discovery. For this OncoAlert academic recap, I'm pleased to present some clinical studies recently published in the journal. First is a small molecule reacts with the p53 somatic mutant Y220C to rescue wild-type thermal stability by Keelan Guiley and Kevon Shokat. In this study, the authors identified the first covalent compounds that selectively react with the somatic cysteine and p53Y220C to restore p53 thermal stability and target gene activation to wild-type levels. Structural analysis also revealed a novel conformational state of the p53 DNA binding domain that may serve as a potential druggable pocket for future drug development efforts. Next is efficacy of a small molecule inhibitor of KRAS G12D in immunocompetent models of pancreatic cancer by Samantha Kemp, Robert Vonderheide, and Ben Stanger. Mutations in the KRAS oncogene are found in more than 90% of pancreatic ductal adenocarcinoma, with KRAS G12D being the most common. Here, the authors tested the efficacy of a small molecule KRAS G12D inhibitor, MRTX1133, in PDAC models with an intact immune system. MRTX1133 prompted deep tumor regressions in vivo in all models tested, including complete or near complete remissions after 14 days, with T cells being necessary for the full anti-tumor effect. Next is NVL-520 is a selective TRK-sparing and brain-penetrant inhibitor of ROS1 fusions and secondary resistance mutations by Alexander Drillon, Henry Pellish, Jessica Lin, and colleagues. NVL-520 is a rationally designed brain-penetrant ROS1 inhibitor with activity against diverse ROS1 fusions and greater selectivity for ROS1 and greater potency against ROS1-resistant mutations than existing inhibitors. As a clinical proof of concept, NVL-520 elicited objective responses in patients with TKI-refractory ROS1 fusion-positive lung cancers, including 2 with ROS1 G20R solvent front mutations and 1 with intracranial metastases, with no observed neurologic toxicities.